thank you so much for joining us for this special seminar with Bob McIntosh here today. I will start saying that Bob was invited to the McFadden Symposium in South Dakota. And when I heard that he was going to be in the US, I couldn't pass the opportunity to them. Like, Bob, just stop by Ithaca. I have had some great interactions with Bob, and our students, especially our early career scientists, benefit so much from the interaction with Bob that I decided that it was a great opportunity for the Cornell students, faculty, and staff to interact with Bob and get a chance to know him. I learned that Bob was here 50 years ago, and 48. So it has been a while since and it was past overdue, his visit. Bob was born in Gloucester, New South Wales, in Australia, and grew up on a dairy farm. He was educated at the University of Sydney, where he received his bachelor's in agriculture in 1960. And in 1963, he got his master, and in 1969, got his PhD all at the same university. His PhD focused on genetics and cytogenetics of flax moth resistance in wheat. Bob spent his entire professional career at the University of Sydney as a professional officer from 1960 to 63, research fellow from 63 to 73, senior research fellow from 73 to 80, and he was appointed director of the Ross Research at the associate level professor in 1980. He was promoted to a personal professorship of cereal genetics and cytogenetics in 1991. Some of Bob's major achievements include contributions to the collection of aneuploid stocks of Chinese spring wheat, chromosome location and genetic linkage studies in wheat, the naming of 7 genes for resistant to leaf rust, 14 genes for resistant to stem rust, and 3 genes for resistant to strap rust. Now you know why I'm a fan of Bob. As a rust pathologist, it's a beautiful career, and so much more he has given to us. Since 1968, he coordinated and published the internationally accepted Catalogue of Genetic Nomenclature of wheat. Bob also designed and conducted appropriate mutational analysis that demonstrated that one gene confers resistance to pathogens that incited 2 different diseases. He was telling the students about that, so I told him to keep more of the story for later. From his genetic studies, he developed white-seeded derivatives from red-seeded alien translocation stocks, enabling the commercial exploitation of LR24 and SR24 resistant alleles in Australian white-seeded wheat cultivars. He did all this without all the genomic and genetic, resources that we have today. So, and the list goes on of all his achievement and resources that he had created for our community. Bob has received many awards from his contributions to agricultural science, including the Farrer Memorial Medal for Service to Agriculture, the Daniel McAlpin Lecture Australasian Plant Pathology Society Conference, Medal of Australian Institute for Agricultural Science, Fellow of the Australian Institute for Agricultural Science, Fellow of the Australian Academy of Science, the G.C. Walker Memorial Lecture in University of Wisconsin, Fellow of the American Phytopathological Society, the E.C. Stackman Award from the University of Minnesota, the Centenary Medal awarded by the Australian government for service to Australian Society of Science in Genetics, Order of Australian for service to agricultural science in Australia and international through research in the areas of wheat genetics and rust disease as an educator and mentor. He's also a fellow of the Australasian Plant Pathology Society. He received the Wheat Warrior Award from the Crawford Fund, and also the Tianfu Friendship Award from Sichuan Province in China. In 2000, Bob officially retired from his permanent academic position at the University of Sydney and was appointed Honorary Professor in the Plant Breeding Institute. In the same year, he was a visiting professor in the Kyoto University in Japan, so that's why you retired, so you can do more work, no? And currently, he's an Emeritus Professor at the University of Sydney in Plant Breeding Institute in Kobiti. Since 2009, Bob has been an instructor for the Borlaug Global Rust Initiative Annual Rust Training Workshop in Njoro, Kenya, spending anywhere from 10 days to 2 weeks engaging with young wheat scientists. For many years, he also reviewed and edited and improved all the abstracts for the posters and talks for the BGRI annual conference. From 2008 to 2016, Bob served as a member of the External Program Advisory Committee of the Durable Rust Resistant in Wheat project that is led by IPCals, where his sage counsel has always proven illustrative and sound. It was at the rust training in Kenya in 2009 that I met Bob for the first time. I was a young pathologist starting working on stem rust of wheat. And since then, we have interacted formally and informally many times. And I've always come out of the conversations in awe. I always learn something. I always get inspired or get more questions. So like I was telling you, I wanted our students to have the opportunity to get a sense of that. Ronnie Kaufman is not able to be here today, but he wanted me to be sure to extend his warm welcome to Bob and to thank him for being such an integral part of the BGRI team, inspired by Dr. Norman Borlaug, which has been working to improve wheat durable rust resistance and climate resilience for the last decade. Bob's input on a professional and personal level has been key to the successes of our project. And for all your hard work, your support and commitment to the Borlaug Global Rust Initiative, and to the wheat community, we present you today with the Norman E. Borlaug Award.
Thank you, Maricelis. Good afternoon, everybody. Well, as she said, this is my second visit to Ithaca. It was 48 years ago when I was here. I don't remember much about it, but I do remember the waterfall, I think, and Watkins Glen, which is nearby, because my kids remember that. But the kids didn't appreciate Grand Canyon compared to Watkins Glen. But I guess there's a difference in magnitude. I'm here today to, well, first of all, I've got to say thank you for the invitation to come back to this part of the world. It's a part that I don't get to very often, even when I do come to North America. This lecture, you can see it's changed title. There's been a mutation somehow. That the title that was offered was the one I presented a few days ago. I didn't mind if I presented that, but we had a debate and she decided to go with this one. So if there's time, we might do part of the other one as well, but that's up to you. You're sick of me before you get there. I'm going to have a few introductory comments. I want to talk about a relationship, a linkage relationship between a gene we call LR13 and hybrid necrosis. And there's a little bit of serendipity in that I'll talk about. And that brings me to other hybrid lethalities in wheat that breeders cope with. I thought I'd just change it a little bit for this audience, for this one to get away a little bit from rust, because people think I lust for rust, but I have other interests as well. I'll talk about some of the allelic relationships we're working on, maybe mention the temperature sensitivity gene and how I'd like to clone that for more physiological studies. And I think Dr. Staples probably remembers some of the classic work that was done by Daly in Nebraska with SR6, and most people have forgotten all that great work that was done at that time, and I think it's nice to reflect on some of that. And hopefully there'll be time for discussion, because this is where students can benefit by asking questions that might be on their mind, and maybe my few years of experience might help out a little bit. Anyway, we'll see. Got to see how to drive this now. Forgotten already. Does that do it? Yeah. We're back. Okay, a little bit about LR13 and hybrid necrosis. This was a system that was described by Hermsen in 1963. It was actually studied earlier by Ralph Caldwell in Compton in 1943, and by Macmillan in Australia in 1936. When I entered university in 1956, he was actually the god professor of our faculty, the University of Sydney. At that time we had one professor of agriculture. Now we've got about a dozen, but that's how things have changed. Now this hybrid necrosis is caused by complementary genes, one of which is on chromosome 5BL, which is just the long arm of the chromosome 5B, the others on the short arm of chromosome 2B. And these genes are multi-allelic, so that different combinations from one with the other, they're complementary, so you have to have both genes to get the phenotype. I'll show a picture later. And they have different dosage effects. So if you have a strong allele crossed with a strong allele, you kill the F1 plant. If you intercross weak alleles, then you only get a moderate degree of chlorosis. And the symptom, as Macmillan called it, is firing. It's just like you put a blowtorch over the plant, which is a good description, but it was later formally called hybrid necrosis. It's temperature sensitive, particularly the ones that are not complete death. At higher temperatures it seems to survive, and there's a relationship there with some of the disease resistance genes, and that's what I'm leading to. One allele of NE2, which we call the medium allele, appears to be the gene LR13, and that's the focus of the next couple of slides. A little bit about this gene LR13, because it has a history in its own right as well. Leaf rust is the disease. It was originally classified as an adult plant resistance, that is, you couldn't see it in a formal seedling test in a greenhouse. Now, what is the nature of a seedling test in a greenhouse to a plant breeder or even a geneticist? It is really an assay to what you might want to observe, or what you do observe in the field. I mean, in agriculture, you're usually not interested in this seedling response. So the experimental system, you're really using a rapid assay to measure something that occurs in the field. And that test may be a good test, or it may be only approximate. Now, those genes that are effective in the field, and you can't see in a standard seedling test, you call an adult plant resistance, an APR. So there was a lot of discussion between us and the North Americans as to whether this was a seedling factor or an adult plant factor. And we saw it as a seedling test, as a seedling factor. But it took quite a number of years of work to decide whether we were looking at a different gene to the North Americans, or whether we were looking at it in a slightly different way. And that took a few years to evolve. So one thing we did discover, that when we made crosses with certain varieties, between varieties with this gene LR13, we would see this progressive necrosis. And if we made crosses with a variety called Spiker, the F1 plants would be dead, would quickly die. If we made crosses with another variety called Line E, we would get a near normal F1 and see some segregation in F2. And indeed, if you cross LR13 into Chinese Spring, you'll see a very, very weak effect if you look at that. So the exception to all this correlation was a variety called Caliansona, which was one of the Green Revolution varieties from Mexico from the 1960s, 1966. And we still haven't fathered out whether that NE2 allele there is really the same as in the other varieties, because it doesn't carry LR13. We made mutants out of LR13 stocks, and most of them were also mutations or all of them, I should say, were mutations at the NE2 locus when you did these assays. In a recent paper from China, they described a new gene in wheat. They called it ELS, in a segregating F4 line. It was an early lesion necrosis. They saw it in F4 line, and they interpreted it as a new mutation. I had to edit this paper for the Crop Journal, which is a journal published in English from the Academy of Agricultural Science in Beijing, had a picture. And immediately I could see that it was progressive necrosis, just from experience. The only exception to that, and I'll show you a picture in a moment, was that it could be barley yellow dwarf virus, which is very common in China as well, or I could be downright wrong. But remember, it was in an F4 line, it's segregated in a 3:1 ratio. So they gave it a new name. It's not in the gene catalogue yet, but maybe it'll have to be a synonym or what, I haven't decided, because this paper's only just been published. They located this ELS locus in chromosome 2bs at the position of LR13NE2, and the photograph was, as I said, was suggestive of hybrid necrosis. So I arranged with this guy saying, well, you know, I think your gene is something we've known about for 50 years. I'm coming to China, can you show me some plants? So fair enough, it was winter wheat, but by the time I got to China, they had the plants. And there was the firing symptoms. And I said, yeah, okay, this is what it's about. So that part was fine. And they had a sequence for this gene. So when I saw the paper first, I actually plagiarised the sequence and sent it to a graduate student. But I didn't tell him where I got the information, because I'd done some plagiarism, right? So he found it was the best candidate that he had for LR13 from the mutants that we already had. So that's sort of where we are and what we're interested. But I think it's quite an interesting story that the first work done with all the test crosses with spiker and line E was done by a student in 1984. The thesis was in 1984, never published, but thoroughly good work. And if I had been sensible enough, she also discovered LR34 in that work as well, but that's another story. So why the single gene segregation if I'm talking about complementary genes, right? So they saw it as a single gene. Well, they were working with an F4 line. They didn't tell us what happened in F3 or F2. So by the time they got to F4, one of these two genes was fixed. The other gene was segregating. That's not bad genetics, Mark, is it? Does that make sense? That'll do, right? Mark has agreed that that's all right. So that's where they got it wrong, and I've given you the genotype there. That F3 line, from which the F4 was derived, would have been homozygous at the NE1 locus and possibly or hopefully segregating at the second locus. Okay? The parents would have been normal, which they didn't see any necrosis, and I guess they didn't look hard enough in the F2 to see the sick plants. All right? I know how operations sometimes work in these exercises while you're making a population. You don't always look at the plants while in close proximity to them, particularly if you're sending a new graduate student out to look after them. So we don't have the data for the earlier generations. So what are the next steps in this exercise? The next steps, of course, are to get the hands on the parents and actually show that one of them, by my prediction, will have LR13. They are winter wheats. The guy gave me the seed of the necrotic plant. Now, if you know something about the Australian quarantine system, yes, I had the right paper to bring the seed into Australia, but it's got to be inspected by pathologists. And when they see these sick plants, I'm going to have to convince them that this is a genetic disease and not a pathological disease. And that may be difficult. I don't have the parents, but I've arranged for these parents to be tested at a laboratory in China. And that laboratory is somewhat south of Beijing, but I'm not sure that they can detect the LR13 gene. So we've got a little bit more to do in this sort of game. But to show you some of the symptoms, on your right, this is the picture from the publication. This is pictures of progressive necrosis over here using spica as a parent where the plants in the field virtually die. Sometimes in a greenhouse at warm temperature you can get a few seeds from those plants. So that gives you an idea. I think there's another picture, is there? Yeah. These are some of the pictures from our publication. This is the 1984 PhD thesis. This would be a spica type cross. This would be more a line E type thing, the intermediate reactions that you can use to classify it. And the pictures in the next one shows you some of the symptoms of this LR13 gene when it's interacting with some other genes in the background. But this is the sort of seedling reactions we get across a lot of different genotypes. They're just two different races of rust. So what you would imply there, that on your left there are some genes present and perhaps interacting with LR13 that are knocked out by the rust strain used on your right. Now, having said that, and hopefully gotten you interested in these lethality systems, it's not the only one you encounter in breeding, particularly when you come to wide crosses. There's one called red hybrid chlorosis, and this is interaction of genes on chromosomes 2A and 2D in common wheat by Emma crosses. By Emma here I mean cultivated Emma. And in fact most bred wheats have the gene on chromosome 3D. And if I go back to my academic grandfather in Australia, he had a lot of problems of crossing a variety called Carpley Emma with good rust resistance with bred wheat. Eventually he found that if he crossed it with a South African variety called Steinwedel, he could get a normal F1. And then this Carpley Emma not only has rust resistance genes, it has powdery mildew resistance genes. And goddamn Briggle, who worked on powdery mildew resistance in Beltsville, crossed Carpley with a variety called Chancellor, which was used as the genetic background for powdery mildew resistance genes. He got a normal F1, and I only know 3 varieties in the world that will give a normal F1 in those crosses. So that's how lucky you can be. I mean, Briggle will say, what's this progressive necrosis? Never heard of it. And yet we have very good records of it. So that's how life goes on, right? And the third variety, I think, was called Gara or something like that. But quite an interesting system. So if you're interested in Emma, you might run around, might encounter this, and you're crossing it with bred wheats, you might encounter this hybrid necrosis thing called red hybrid chlorosis. There's another system that when you cross Emma with Timofeevi, it's got another name that's given there, CS1, and the chromosomes are given there between the interaction of the genomes of Timofeevi and so on. So we talk about wide crosses in wheat, but it's not all smooth sailing, right? You may have to do bridge crosses. There's another system at the bottom that involves Igelops tauschii crosses, and earlier with the students we were talking about synthetic wheat. Not much in the literature about that one, but the Japanese encountered some problems with this Langdon germ being crossed with Triticum tauschii. And there's one gene on chromosome 7DS, that's the tauschii gene, and the other gene, to my knowledge, is unmapped. Here we have another system. It was interesting when I was in China talking with the guy on the hybrid necrosis. He said, oh, he just had a letter from a friend somewhere in China saying that he had this strange dwarfism. And of course these days I said, can you get me a picture? So he sent the telephone message, and within seconds it was back with a picture. And I said, grass clump dwarfism. You know, this poor guy, I'm sure he had the idea he discovered a new system of dwarfism, but you know, this was all worked out in the 1930s, basically. Again by Macmillan, who was the professor I referred to earlier. He called it grass clump dwarfness. But by Hermsen in Holland, who formalised it genetically better later, it was called grass dwarfness or grass clumps, another term. I guess you've encountered it, Mark?
I think so. But
if you cross a West Australian variety, you'll get it.
Unconfirmed, but I've seen them before.
And the interesting thing about that is you can get an F1 or you can get it an F2 because it's a complementary gene system, and I'll go through some of the genotypes. You have these genes are called D1, D2, D3, D4, D2 and D4 on homologous chromosomes, and they are duplicates. Whatever you can do with D2, you can do with D4. Now, if you have one dose of D1, one dose of D2 or D4, one dose of D3, you'll get a grass clump in F1. If you don't have the D3, you'll get a normal F1 and you'll get segregation in F2. Now the D4 gene, as far as I can tell, is relatively rare, and the ones which have been described as Capelle de Pré, a French variety, Cheyenne, which is a Nebraska variety, and Brevard, which is an old classic winter wheat, I guess, that was the basis of some of the breeding in the Pacific Northwest at least. So there's some dwarf genotypes there that you can get in F2. If you've got 2 doses of D2 and 1 dose of D1, you'll get a dwarf. But you can only get that in the F2. And the picture shows these little, not the best picture, but it's one we could get our hands on. I can tell you the Chinese picture was much better because it was nice and green. So that's that story. So my warning there is really rediscovery can occur and does occur. I want to now tell you what I call the Gabo story, because it's interesting. It's a bit like this seminar. We weren't quite sure of the title, right? There was a variety was later called Gabo. It was Bobbin 39 by Garza, and some guy from Australia brought 2 lines to the US in the 1930s and got them mixed up. So this particular Gabo sib was called Timstein in the US, but it was really an Australian cross. The variety Gabo in Australia was the foundation variety for quality segregation of wheat many years ago, but was bred for rust resistance. Had the gene called SR11, released in 1945, susceptible about 1949, so I know it's the old story, a breakdown. But Gabo is the same as Timstein. One thing that happened there when they located the resistance to stem rust in Timstein in North America, they used the crosses with Chinese spring, and they got strange ratios. They got a ratio of about 2.4 to 1. And when you get a ratio like that, you can explain it by having 2 linked genes. I mean, you could explain it theoretically and make an estimate of linkage. So the genes there were called SR11 and SR12. And in Australia, they were making crosses between this material and a variety called Federation, a very significant Australian variety, and I might refer to it in a moment. And they were getting normal 3 to 1 ratios. So this argument came up as to whether it was 1 or 2 genes. Well, eventually it was decided that it was 1 gene. When you made crosses with Chinese spring, there was some effect that Chinese spring affected the ratio. And we know now when people do molecular studies, they find a lot of sites in the genome where they don't quite get the right ratio, and they usually chuck those guys out and only look at the ones that segregate normally. But that's indicating that within the genome, there are factors that drive segregation. If you get stuck in there, in those regions where you're looking at a real gene instead of a bit of DNA, then you'll get bias ratios. And Sears and Legring, I guess, called this killer factor. They had evidence that there was a killer gene, killed the pollen, and you got a biased ratio from that. But it was the work done by Lewig in Australia was much more involved, because he was able to show that different sources of different varieties with SR11 crossed with Chinese spring would give you different ratios. Slightly variations on that ratio. And he called it differential transmission of gametes. But I suspect it's just the same thing as the molecular people find when they find these areas in the genome of distorted inheritance. So there's a gene there that causes distortion. As you walk away from it, the ratio dilutes into normal segregation. I certainly would like to see more study of that sort of thing. So that was the SR11, SR12 story. Gabo, or Tim Stein, also had leaf rust resistance, and this was based on where you worked and which races of rust you worked with. Now, I'm talking about stuff coming out of the 1950s and 1960s. But eventually, there were two genes identified. One was called LR23 on 2BS. That gene is not the same, but very close to the one I had a moment ago, LR13, very close in the genome. And LR10, which is on 1AS. Now, at that time, when I came from my postdoc in Missouri, I brought a lot of old Australian varieties, sourced from our famous original breeder, a guy called William James Farrer, who came to Australia in the 80s because he couldn't stand the climate in England because he had consumption, tuberculosis, if you like. He went to a drier environment. He was a mathematician. He was at Cambridge University at the same time as Darwin, but I can't say they went to the pub together or anything like that. But it was about the same time. But he was a surveyor in the countryside, just a land surveyor, and he saw rust in the field, and he saw the farmers' problems with rust. And so, by observation, he became a plant breeder, a private plant breeder in the first case. And he had established contacts with people in India, in England, and in North America. Saunders, who worked in breeder of Marquis in Canada, I guess, and with Blount, who was a breeder in Colorado. And on one occasion, you know, there was no internet in those days, as you know, and not even aeroplanes, right? And he made his crossing forceps from his wife's hairpins. And he actually sent his crossing pins, I think it was to Blount, because they were discussing the tools they used to make crosses. You know, and this went by sea mail, you know, whatever, 6 or 8 months later it comes back, if it did come back. You know, things, you had time to think in those days, really. But it's an interesting story how these people established their contacts. And this is very similar to what we were talking about just 2 days ago about McFadden in South Dakota. But McFadden came a little bit later than Farrer. But Farrer's wheat, he had many arguments with. Still have arguments. Did Farrer breed for rust resistance, or did breed for rust avoidance? Because what Farrer did, you know, these Englishmen, when they colonise, do some stupid things. They come to low latitude countries and they try to grow English varieties that respond to day length, right? So you get a bit concerned that when the wheat ripens in the middle of the drought in late summer, it's not very good stuff, right? Because it's not adapted. Farrer introduced wheat from India and from South Africa and crossed with the fife wheats out of Canada. The fife wheats would have been day length sensitive but had quality. And for the students, do you know how they tested quality in those days? What was good quality wheat? You chewed it up and made a nice big golly in your mouth, and if it stayed stable, you know, that was a sign of strength. It was the chewing test, was about the only test they had till about 1916 or so, when I think mixograph was invented about that time. So you had to be a good chewer for quality. So, but Farrer knew that he wanted quality, and he decided he wanted the day length insensitivity. The day length sensitivity moved the wheat growing areas in Australia to the drier areas. And we know that rust needs moisture for germination, so rust avoidance was there. But I brought some of Farrer's varieties, a number of them, old Australian varieties, to Missouri in 1970 because we were interested in this Gabo story, and the bobbin goes back. It wasn't really bobbin, it was probably another variety, but it's got an accession number, that accession. I brought them to Missouri and I tested them with leaf rust, and they all had LR10. We'd never seen that gene in Australia with Australian isolates of leaf rust. So did Farrer really breed for resistance to leaf rust? I believe he did. Then formal studies of rust in Australia started in the 1920s. And the first professor of plant pathology at Sydney University was a Scotsman who got the chair, but on the way to Australia, he stopped off at St Paul, Minnesota and visited with a man called Stakeman. So brought all differentials and so on, and he picked up the leaf rust differentials from North America at the same time when he did the tests. There was no LR10 there. He couldn't see it. And I would think, looking at the literature and what happened, that Farrer bred for resistance. And then we had this very important variety called Federation in the year 1900 that was released and was grown everywhere. The rust attacked Federation, the virulence became fixed in the population. So by the time Waterhouse got there and looked at it, the A virulence factor was gone. It was all virulence. So it was never seen, but it was in his varieties. There's a stem rust resistance gene that's in those varieties as well. He probably put there and was never discovered in Australia. So when you move internationally with materials and pathogens, there's a lot to learn. You can often get caught out because of the genotype of the pathogen in relation to usually the fixed genotypes of the host that you have to work with. So that was a thing to learn in that was come from the postdoc work that I did that time. I never published it, but it was just interesting stuff. So that gene was floating around and may originally have come from Farrer as a material. So the last sentence just formally says what I've been saying. Now, when gene cloning became a possibility in the mid-90s, and I guess we were talking about this for some of the students and postdocs earlier, I was asked which genes to clone. Which genes do you think they could clone? We know we've got dozens of genes out there. Which ones should we target first? And I said, SR2, which is the McFadden-Hope resistance, which has been durable for such a long period. So I selected it because of its durability. Or recommended it, I guess, is a better word to say. The other one was this system of LR20, SR15, PM1, a linkage in chromosome 7AL that was never broken in genetic or germplasm screening studies. So what I mean by germplasm screening in that work that went from the 1930s to the 2000s was that you test with one pathogen, test with one for one disease. If you see the resistance gene, you ask the question, well, is the other gene there? And you can do it from the direction of rust as the initial test or mildew as the initial test. And in all cases, the three genes were present. So that's a bit like GWAS, I guess. That's what you're looking for, things that hold together in a biased way. I don't see it as any different to that. It was the way we did it in those days. Mutagenesis of that system revealed that there are probably two genes. The leaf rust gene and the stripe rust gene mutated together. The mildew gene was independent. Now there's another pair of genes that I believe are pleiotropic in the same way, and they're known as LR16 and SR23 in chromosome 2BS. I think I have a picture just on that in a moment. So the first two are the ones I recommend to cloning. You know, now we've probably cloned that I know of about 10 genes for rust resistance in wheat, but we still haven't cloned the LR20 PM1 system or the SR2 system. So I might have been lucky earlier with necrosis, but I wasn't very lucky with this recommendation. But you know, life has luck sometimes, but frequently you miss out. Some time ago, we published that we thought the genes YR5 and YR7 for stripe rust resistance were allelic. Now, some work in England in cloning these has revealed that they're probably not allelic, but extremely closely linked. We haven't got crossovers, but they're not the same genes. But it's interesting that Another gene that comes from Spalding's prolific, which is an old European variety, seems to be a truncated version of YR5. That's where that's worked up. We have another gene at that position. We call it YR-Jub, J-U-B, for a Bulgarian variety whose name I can't pronounce, so I won't try. We just call it YR-Jub. We think that might be at the same locus. So that's interesting. The other one I'd recommend for target, or have recommended for targeting, and we've got candidates, is SR6, which is a classical temperature-sensitive gene. And I see the temperature sensitivity as being useful for later work on functionality and so forth of the resistance gene. At present, we're targeting the SR9 allele series for stem rust resistance, but in Australia we can only see SR9B, SR9E and SR9G. And hopefully the other two listed there, maybe the Minnesota people will concentrate on those, because SR9A and SR9D, which is one of the two genes in Hope in the Hope variety, we can work together and look at an allelic series. And if we can understand how an allelic series work, it may help us in designing genes for the future, but I'm not so sure. But it's worth a try. This is our attempts to clone, or the people I'm working with basically, than my attempt to clone SR2. As I say, it's not cloned, but we're down to 2 candidate genes, but it's not all clear. And you probably know that when you're doing experiments in containment facilities, it's sometimes very difficult to do the real experiments, what you want to do, like infection studies and so forth, because you've got to build up everything into your containment facility. So the variety that's used for transformation, or one of the varieties that's used for transformation, is the Pacific Northwest variety Fielder, and this is a picture of a Fielder without SR2. This is a picture of Fielder with SR2. And there's 2 candidate genes. This is a back-cross derivative Fielder with SR2. You see, it's not easy to classify. That's the first problem. In this type of test, which is a monocyclic test compared to the field, which is a disease progression test. You can't put these plants in the field, at least not in Australia. Maybe we'll be able to do it in China, I'm not sure. When you've got both candidate genes present, it seems reasonable to say it looks as if it's got the resistance. I don't think you have to be a wizard to suggest that. This one's fairly clear. These are not so clear. If this first candidate is missing, not much joy in that. And the second one, there's a little bit of necrosis up here, but it seems both candidates might need to be present. And that's about where it's up to. So it's not a particularly good story, but after 20 years, it's a damn good progress. Now, I was talking about LR1623. I want to just show you that the classic LR16 phenotype, a very necrotic pustule. Right? These so-called, for the students mainly, so-called resistance factors or immunity factors that we work with in our crops are not total immunity. They are really intermediate responses, and it is the hypersensitivity or the necrosis that leads to the resistance you see in the field. Okay? And that necrosis does not kill the fungus. It contains the fungus. And temperature work, I mentioned earlier, I think the work in by people in Nebraska and in Canada, that with gene like SR6, you can do differential temperature studies. So you can induce the necrosis in a SR6 interaction by low temperature, increase the temperature, and the fungus will go out and give you a uridial circle around the necrotic tissue. So the fungus is still there, it's just contained. You give it the right conditions and it'll grow. This other one is this gene here. In leaf rust, it's very easy to classify in a in a single gene line. But CIMMYT materials mix up their leaf rust infections, and we were using this phenotype as an assay for that. Now, no way can you imagine this is a resistance gene. But see the necrosis, the dead necrosis there. So we were actually using this phenotype with stem rust as a marker to predict that gene. Now we saw it through thousands and thousands of screenings. We could see this relationship. So I'm postulating that the gene that gives this effect also gives this effect, and you can call it SR23 or you can call it rubbish, whatever you like, but it's a phenotype that you can see and you can classify. We have one race of rust where the phenotype is significantly less than that. It's an old rust race that dates from the 30s. So, you know, this is just using markers. Markers are not new. We've been using markers for well, SR2 and the black chaff. We've been using them for nearly 100 years. So it just takes the concept from phenotype to DNA. Going back to the SR5, SR7 story, this is the sort of phenotype one gets for YR7. The Y's there and the R's down there, something wrong with the way we set it up, sorry. And these are YR5, YRSP, and this is the Jube2 reaction with a particular strain of Puccinia striiformis, the stripe rust pathogen. Now, one thing that I'm entertaining is that I've said that this gene and this gene we now know are not allelic based on the cloning work. Recombination-wise, they were allelic. These, we think, are allelic. I said this is a truncated version of that, but you've got races of rust that can attack that and not that, and vice versa. Only in Australia, by the way, for this one. And we have this one, which we know less about, which is another specificity. What I query is whether this difference here is enough to postulate those genes are not alleles. It's just something I'm entertaining, that whether the actual symptom you see would be indicative of allelism in this type of system. I mean true allelism here. Now, I make mutations for rust resistance, but I also love looking at the other mutants that turn up in the population, right? This one is a variety. It's a Triticum compactum, club-headed wheat. It's a single gene on chromosome 2D. It has SR6 in it. SR6 about 20 centimorgans from this gene club. So you grow out a mutagenized population, and these are some of the things you see in that mutagenized populations. I don't know what's going on. It's more variability than I expected. This is a single gene effect relative to that in genetics, but we're getting these different types of spelts. This one's got a long spike. This one's got a very stiff spike, so it's interesting for the understanding the spelt system. And I have another one of that, similar to that, but we've got these mutants. They're just different plants in the populations. You get all this variation. The other thing you get in mutagenized populations is this effect. And Ernie Sears called this corroded many years ago. Back in the probably 1940s, he published a paper. It can be caused by genes on the group 6 chromosome, 6A, 6B, 6D, and the gene is called CO, and you can name it 1, 2, 3, or you can name it another way. There is a South African variety called Kurichi that's regarded as a variety. It carries this corroded factor. They're duplicate factors, duplicate genes. I saw a lot of this when I was working with aneuploids in my younger career, turn up in group 6 aneuploids. but it was random across the three. When you made different crosses, you seemed to pick them up in different combinations of those chromosomes. This is another mutant here that's chlorotic. It's not the same as that. And here it is again, I guess, this guy. Now, this is interesting because we grow the plants on and this phenotype gets lost. It's a juvenile phenotype. And in rice, there are many genes. And in humans, there's many genes expressed in juveniles, but they're not expressed. Once the plant or the animal grows a bit, those genes are never used again for some reason. There's a lot of these mutants also in rice. They get chlorophyll mutants in rice and the plant grows out of them. But they're mappable genes. They find all sorts of effects for them. And there's many people working in China on these things and think that those genes are important. I'm not sure they're so important, but they're mutants that you see. Continuing with mutations, just for excitement, this is the corroded. This one here speckled. This one we call scorchy. It looks more burnt. It's certainly phenotypically different to the other compared to wild type. This is very common in mutant populations. You get these short, fat leaves, and I think a physiologist could look at that and do something with it as well. Sometimes it's almost lethal. I call it onion leaf. And then another version of it, which is probably the same gene but not to the same extent, is just shorter leaves than normal. So all of this exciting stuff turns up. Now, a student of ours who's been working in the lab, just about finished the PhD, has tried to visualise where we've come in cloning genes. So she has two cartoons that I'll share with you, and I think it's quite clever, I think. How do you clone a gene from the wheat genome? And she's starting by what she calls the traditional way, a large segregating mapping family, and she's compared that to a lake. You go find the lake. Which one are you after? Which lake are you going to fish in? So you do some genetic mapping from wheat, or comparative genomics, and you try to figure out in which area a particular fish might be located. And the only thing you can do is change, to find the target area, which chromosome she's saying. Then you do the physical mapping with back libraries or reference genome, and you use a rod to catch a particular fish. The only thing you can do is change the bait. Right? So down here it concludes you're doing candidate analysis. Down here she says, well, it's not uncommon to have 2 or 3 PhD people to catch one fish, right, or to clone one gene. The next picture tries to update this, because this is what she's done. So she's using MutRensec, mutagenesis, R-gene enrichment and sequencing pipeline. She's got the publication there. So you're going to use the wild type and mutants. So you get to know the fish you want, right? So the fish we want in a lot of disease work is NBSLRR, that's there. So you need to make a net to catch the fish. The portrayal now is you can catch these fish in bulk. So you use the NBLRR type gene as baits, it's a library, and this comes from sequenced genomes. You fish out all the R genes from the genome of the wild type and mutants, so you get all the fish out of the lake by using a fish net. Right? The next step is R gene sequences from wild types and mutants are compared. This is sorting the fish by machine. Identify the potential gene candidate. The computer will give you the fish you want. The conclusion is it's possible for one PhD student now to clone 2 to 3 genes. So that's progress, I guess. So for this talk, some acknowledgements, which is interesting. I spent my whole career, 40-year career, on soft money. So if you've been on soft money for a while, don't get concerned if you do a fairly good job, or you think you're doing a fairly good job. There is hope. I use that word hope quite a bit, don't I? With a capital and a small letter. There's been lots of open discussion with colleagues in many countries. This is becoming more constrained in my experience with greater privatisation, the level of breeding, and with greater competition at the level of research. I don't know, it's getting a little bit more. I'm a bit old, I can't always make PowerPoints, so Pung helps me with that. And I came here under the support of first the Dakota State University and this university. So thank you for that. Any questions?